So, good afternoon, everyone. Welcome to the second webinar of 2023 H3D Foundation webinar series. My name is Shruti Singh, and I'm thankful to the speakers for accepting the invite and participants for attending the webinar. Our today's speakers are Dr. Christian Zeine and Mr. Nikhil Rautela. They represent US Pharmacopeia. The US Pharmacopeia works to strengthen the global supply chain so that the medicines people rely on for health are available when needed and work as expected. So before we start with the talk, I would like to let you all know that we have a Q&A session at the end of the talk. So questions can be posted in the chat or kept for the end of the talk. And today's topic is value of pharmacopoeia in early drug discovery, research and manufacturing. And now I would like to request the speakers to enlighten us with their work. Over to you.
Can't hear you, Christian.
Sorry, I was on mute again. You should be able to hear me now. Can you hear me?
Yeah.
Yes.
Yeah, we can hear you, Christian.
All right. So let's go then to the presentation. You should also be able to see my screen now. Confirm?
Yes.
Very good. Yeah, I see you nodding. Okay. So good day also from my side. My name is Christian. Thanks for the quick introduction, Shruti. And my name is Christian Zeine, and I am the Scientific Affairs Manager of USP for the EMEA region. That means Europe, Middle East, and Africa. And I'm working with USP since 2019, currently sitting in the office in Basel in Switzerland. And I think with me is today Nikhil. And Nikhil, please introduce yourself as well before we start on the presentation.
So good afternoon, everyone. Thanks, Shruti, again for the invitation. So my name is Nikhil Rautela. I'm a colleague of Christian Zeine. I'm in the EMEA office of US Pharmacopeia. I joined USP in 2021. And my focus is mostly on the biologics part of US Pharmacopeia, that is monoclonal antibodies, vaccines, cell and gene therapy, biomarkers, clinical biomarkers, and peptides. So we look forward to today's talk. I'm happy to be connected with UCT again. I am a UCT grad myself, having been in Cape Town for 4 years, and I'm aware of H3D and UCT quite a bit. So happy to be back in UCT again. So Christian will start the presentation, and then I will follow up with my section towards the end. So all yours, Christian. Go ahead.
Okay, thank you. And while Nikhil is more specifically the expert on bio, I am more an expert on small molecules with a focus on reference standards and impurity testing in the area of small molecules here. And I will take you now through the first slides of our presentation here. So let us have a quick introduction to USP first. So we are more than 200 years old. USP was founded in 1820 by a range of 7 doctors at that time, 7 physicians, and still is a non-profit, private, independent, and self-funded organization institution here. And we are value-driven and focusing on empowering our staff and our volunteers on the setting up of quality standards for medicines and the APIs, the drug substances, and excipients in there. We do that with more than 1,000 employees worldwide. We have laboratory facilities in the United States and in India. There's also a small laboratory in Ghana, but that is not producing reference standards. And we have also offices in other parts of the world. I am right now sitting in Switzerland. We have also offices in China, Brazil, Singapore, Bangladesh, Ghana, Ethiopia, and also Nigeria. So you see, we are internationally focused, globally focused, and we are also internationally recognized. Also, when you look into the regulation of a lot of the health authorities in the world, including SAPRA, for example, in South Africa, we are recognized there as one of the official pharmacopoeias that in a way is when you show compliance to the USP, the drug products, but also the APIs before and the excipients, they get normally the nod from the regulatory authorities, and you can proceed then there. So that's and we will see in a bit how we are doing that today. We are seeing that today how we are doing that in the United States Pharmacopeia. I mentioned already that we have 1,100 employees worldwide. Much more important and relevant are our 900 scientists, practitioners, or even more than 900. We are close to 1,000 there nowadays for the scientists, practitioners, and the regulators that we embrace as volunteers, and that are in the end acknowledging and releasing the standards, the quality standards into the market. So at USP, the staff is preparing all that, is giving the data to the volunteers, but the volunteers, those scientists, practitioners, and regulators, they are deciding which standards go out and are made publicly available to protect the public health here. So that's basically the introduction, and our mission. That is, of course, a given here from what I mentioned already before, but here it is kind of put and condensed into the mission here is to improve the global health through public standards and related programs that help ensure the quality and safety and benefit of medicines and food. And basically at USP, we are looking on the quality, and by that kind of ensuring also safety and then the benefits then here. We are talking today mainly about the public standards and only slightly touch on the related programs, so that you are aware what to expect today from our presentation. So let's have a look first on the white paper on the value of pharmacopeia standards that was set up by the World Health Organization's International Meeting of World Pharmacopeias, where a number of pharmacopeias are meeting under that umbrella. And in 2020, we came up with that white paper here, where you see, we'll see if you look through that these are 8 pages that you can download via the link below there. And you will see there from the text that the pharmacopeia standards are facilitating the preparation and the assessment of regulatory submissions of the drug products, of the finished dosage forms, of course, but also the excipients and the drug substances before, and is providing there a public mechanism for that. Let me see if I can. Yeah, I can do that. Providing a public mechanism to independently verify the quality of a product at any time during its shelf life. If you go a little bit more into a little bit more into the paper, then you will see. I hope you can still see my screen. I had an issue here at them, but it seems it should work still. Yeah. So, and if you look into that white paper a little bit more, then you will see that it is explaining that a pharmacopeia, or how a pharmacopeia, is an official collection of quality standards and specifications for the medicines and the ingredients. And it is important and relevant for all these stakeholders that are mentioned here. So it's basically the manufacturers, of course, but they are dependent to a certain extent on the raw material suppliers, those that are providing the drug substances and the excipients. They also are our stakeholders and find quality standards there. The analytical laboratories that are giving contract analytical laboratories that are given services to raw material suppliers, to manufacturers. They also look into the monographs of the USP, also of other pharmacopeias. You find a collection shown here. So they look into the monographs and use those methods there in order to show the compliance to the specific pharmacopeias. Also, pharmacies, regulatory authorities, of course, like SAPRA here mentioned here, like FDA, our main regulatory authority in the United States and all over the world, where USP quality standards are recognized. They are also looking, of course, into our publications, into our decisions here, and the policymakers, nonetheless, of course, as well. So what is also mentioned in the white paper is that medicines, of course, are complex products made from all the ingredients there, excipients, the drug substance here, and the quality of medicines is then ensured by many factors, but a very important factor is, of course, the quality of their components. And that is basically where the United States Pharmacopeia, where a Pharmacopeia is looking into. Because when you look on the medicines and then their ingredients, they need, of course, to be safe and effective and of a high quality. And by the quality standards, mainly of products that are already approved in the market of the United States, we can then, by providing these quality standards of already substances that have been proven to be safe and effective earlier before with a during an innovation process here, we can also then make sure that they are safe and effective, and then they're also working together when all these excipients are coming together into the medicines. And we provide these quality standards. We also, however, provide also best practices in our general chapters and guidances in our general chapters that can be used even by by innovators and not just those users of the quality standards for already approved products here. These guidances are preferred techniques and procedures to be used in the testing of medicine, and that's basically, as you can see here, these procedures are looking into proving the identity of an excipient of a drug substance, and then also purity. So looking into what impurities are present in the product, and then also looking at the potency at the assay of the drug substance. And these standards, the best practices and those guidance, as I said, are used a lot by generic product manufacturers, but also are important and relevant for those that are looking into innovative medicines and are just at the beginning of their research there. So we have, for example, recently talked to a company that is very innovative in the field of the disease ALS there, and they are looking into combination product for that. One of these products, sodium phenylbutyrate, is that's open source information. I'm not telling you a secret here. So one of these components is described in the Pharmacopeia, in the monographs with quality requirements there. The other compound is not described in the Pharmacopeia, but they are saying we can also use related drug substances and see what impurities are coming up there in that related one, the limits of that impurities, and can draw conclusions for our bile acid, which is used there as well for and very much comparable to what is already in the monographs. And they also said also the general chapters, and specifically dissolution and disintegration for the product that is now in clinical in the clinical phase. To have this information is very important, and that is what we are providing. We are providing procedures there in the general chapters. You will see that in a second. So not only for a drug substance or an excipient, but also general chapters on specific methods and approaches to certain topics in the pharmaceutical environment. So, but basically, with our quality standards, we realize the independent verification of the product of the quality of the product at any time during the shelf life, and by mentioning those standards in the dossier, it's helping also to build confidence among the healthcare professionals, the assessors, and then finally, of course, also the patients. The Pharmacopeia, it's not written in stone. It's not chiseled in stone. It's continuously evolving. You will see that in one slide in a second. And we are looking there, of course, into the science and regulatory environment and are adapting our standards there continuously. Some other publications that are dealing with the use and the relevance of reference standards, sorry, not of reference standards, but of compendial standards. are these two here, where Murimi-Vostell could show that that the pharmacopeias are used by manufacturers, especially here in this case, it was on the example of the USP, and how it is used by manufacturers and regulatory bodies to help ensure the quality of the pharmaceutical products. And they were also showing there that it is facilitating the access to affordable medicine. So what they showed in the article was that whenever a pharmacopeia monograph is there, the competition is broader, there are more generic products on the market and more affordable medicines than they're available. And the other one is from our colleagues at USP, where they showed that compendial standards are of a high value to the pharmaceutical industry by interviews with the pharmaceutical industry that was done. And it was shown that they are decreasing the development effort of pharmaceutical articles in quite a manner. Yeah, I wouldn't say significant, because that is having a statistical meaning here as well, but it is definitely relevant to decrease the development effort of pharmaceutical articles. So as a kind of summary in between here, for the quality standards to be impactful, it's a kind of summary what we have seen on the slides already before. They need to be developed first, of course, by independent experts. That is where we are working with. We will see that in a second when we discuss how a monograph is set up. And they need to be practical, of course, for the users, but also for the enforcers. So we need to see that we are really doing meaningful work here, and are not completely disconnected from the market here, of course. They need to be adapted and approved all the way through technology and the evolution of healthcare. We need to see that we are always not necessarily state of the art, but that we can provide the quality at the necessary level here that is achievable with technology and with the evolution of healthcare there. So, but the methods not necessarily need to be state of the art. So, but nonetheless, we are going from thin layer chromatography, for example, and paper chromatography. Paper chromatography is not used anymore in the pharmacopeia, but we are moving there to the column chromatography, gas, and specifically liquid chromatography, for example, with all the monographs are modernizing them there. That is also very, very important. And of course, they need to be aligned with public health and the patient safety priorities here as well. So, and this is here just a quick showing very in a rough manner how the standards are really evolving. If you look back into the year, into 200 years ago in 8020, the USP was a single recipe book, but it has now evolved into procedures and acceptance criteria to support the medicines in the marketplace. So you see here some snippets from the monographs of heparin and filgrastim, and also a collection of general chapters that are supporting these two monographs in order to set up meaningful and check on testing on meaningfully on the quality of these products. So, and how are we doing that specifically? So when you look into the United States Pharmacopeia, into the USP, you will see that there will be what we are calling documentary standards and reference standards. With the documentary standards, we mean basically our USP publications, where you find those official documentary standards there for the drug substances, for the excipients, for example, and then also the general chapters, to which we come in a second. These are the documentary standards, and very, very often the monographs are linked. Also, the general chapters are linked then with what we are calling a reference standard, where you use that reference standard. That is basically a very well characterized specimen of a small molecule of a biological standard, for example, and you use that reference standards together with the methods defined and explained in the documentary standards there to ensure the quality in development and manufacturing in the pharma area. So you use these together, and we are saying that only the combination of documentary standard and of the reference standard is giving you the conclusive compliance. So it's showing you that that the requirements has been fulfilled in a conclusive way, and that combination is establishing the quality benchmark where you need to compare your samples against there in order to show the compliance to USP, which is then also a big step in order to get material approved in the pharmaceutical market. On the drug substances, on the excipients, but also when you put them together into the drug product, there are also documentary standards for finished dosage forms for the drug products in the United States Pharmacopeia. So this is, and I would like to take you through this process in a little bit more detail. Let's see where we are with the time here. So this is how we are developing, and giving a nice overview how we are developing the monographs, the documentary standards and the reference standards. So at some point, it is identified, there is the need identified that we need to have a new standard for a new medicine, for a new excipient, for example. And we are looking then at the USP stuff. We are looking at industry experts that can provide us, and drug manufacturers that can provide us with their methods in order to set up a draft public standard, a draft monograph proposal, so to say. So we are not doing that. Sometimes we are doing that on our own, but our preferred way is to work directly with the industry partners to get meaningful methods from them, how they check on the quality of a certain drug substance, excipient, or drug product there, and put that information into USP language so that it can make up a draft monograph here. That draft monograph is then reviewed by a committee of experts. These committees of experts, these are the volunteers that I was referring to in the very beginning of the presentation. These committees are looking then onto the draft monograph and are saying, yes, it is good to be published as a draft, and then it's getting published as a draft in our Pharmacopeia forum, and people have then 90 days, the interested public has 90 days to look into that monograph, provide comments to us, and then our staff is looking at these comments, is working on the comments together with the expert committee, and is then hopefully approving the reference, the monograph, the documentary standard at a point. And in parallel, also the reference standard, if needed, is developed here in more or less the same way that we get donations, like we get donations on the methods for the documentary standard. We get bulk material donated by the industry partners and by our sponsors, and we are then setting up a testing protocol that qualified labs around the globe are performing there, and are in that way confirming the candidate's material identity and establishing the purity and an assigned value. So that is also then going, that work is done basically in USP laboratories there. That data, that information is also going to the expert committee that then ideally together with the documentary standard can approve these two standards so that they are then published and used together in the official combination, documentary and reference standard, to give the conclusive combination here. That's basically how we are working on that. To set up a new monograph can take years. There are also reference standards to set up that kind of standard. That kind of materials can take also a year, for example, for setting it up in the as a first standard, as a first lot here as well. Replenishment lots are running quicker. That's also what we need to do once a reference standard has been depleted here. That's also then where we need to replenish that lot. That gets a little bit quicker done nonetheless here. So these are our experts here, the committees that I was talking about, and who are making the decisions for the standards being released. So you can see that they are set up by the different disciplines, so to say, biologics, small molecules, excipients. The general chapter committees are the ones that, of course, are dealing with all the general chapters, not directly with the monographs there, but with analytical methods, and we will see an example for that in a second as well. And we also have committees on healthcare quality and safety, and on the dietary supplements, herbal medicines, and food ingredients. And as I said, approximately close to 1,000 volunteers are working in those committees, and setting up then the council of experts there as well from these committees. A quick look on the documentary standards here and how they are set up. We have under the documentary standard basically three levels, as you can say. So we have the general notices, which are providing definitions for the terms that are used in the monograph, some basic information to interpret all the monograph requirements. Then comes the general chapters there, which are discussing analytical methods, other broader topics, and widely applicable methods, approaches there as well. And they are not linked directly to individual products unless they are referenced in a monograph, and we will see that in a second as well. There are also what we are called chapters below and above 1,000. The one with the number below 1,000 here. The numbers below 1,000, they are enforceable and are containing validated procedures, and are very, very often also linked to reference standards. We also have chapters that are above 1,000. There are normally no procedures in there. That's not completely true, but they are normally not having procedures. They are also providing only general information and are not directly enforceable. But very, very often, those chapters are containing information which is up-to-date information the regulatory authority wants to see as well. And very often regulatory authorities also reference a chapter over 1,000 when there are discussions with certain applicants, for example. So they tell you, you will find, for example, a bit of information how to test on nitrosamines in the chapter 1485 of USP. Have a look there. There are even methods described. And in that way, it can go there with the methods, with the chapters over 1,000. And then we have the monographs, which are very product specific and contain then the test methods with the specifications mentioned there as well. And those are very, very often, almost always linked to reference standards as well. You can see here such a monograph example. I hope you can read it here. So for example, if you check and search in our online version of USP, and by the way, we have only now an online version anymore at USP. We abandoned the printing of a book. I think it was 3 years ago. So when you type into the USP, the word acetromycin, or just as part of acetromycin there, the first 3 letters, for example, then you come up with a list of monographs that are dealing with the drug substance here, but also with finished dosage forms with drug products that you can see here. That is how a monograph looks like. So we have definition given here with a clear statement of the assay, for example, then information about identification. Basically, that is done by spectroscopy, and very, very often by IR spectroscopy, where you have the reference standard there, an IR spectrum taken from the reference standard, an IR spectrum taken from your sample to be tested, for your substance to be tested, and you check then if those 2 spectra are identical, and with that you have then kind of identified your product there according to the monograph. You look then into impurities and then also into the assay here with the methods that are described here and which are linked to the reference standards then as well. There are in the methods, there are system suitability requirements given. So when you transfer the method from the book, so to say, or from the monograph, it's not a book anymore, but from the monograph into your laboratory to see if you have transferred it correctly, you can run the system or you need to run always the system suitability tests there before you are testing on the real samples, and make the results then they are valid. So that is also information that is given there. Very often it's about chromatographic resolution. You see that example here, that the resolution should be not less than 3 between this impurity, which is eluting very closely to the API in this case of acetromycin. And that is always how it is done, that you check, or very often how it is done, that you check the resolution of a closely eluting impurity to the API. And by that, you can assure that also the other impurities are separated in the right way. That is normally how it is working. There might also be some specific tests based on the monographs here, but let's not go into detail on that here. So that's the monographs. There are then the general chapters. You see, for example, here, I'm not sure if you can read it, but here it is saying chromatographic system. And you see then here that this monograph is then for the chromatographic system, referring to the general chapter 6.1 on chromatography, for example. And when you look into that chapter in USP, you find their basic description of the different chromatographic techniques there. I mentioned that we don't have so much paper chromatography anymore in there. There might be still some thin layer chromatography techniques in the monographs here. It's described in the chapter how the stationary phase can look like, how the apparatus should look, how you detect, visualize, and also then specific procedure information there as well. But the majority is going into the column chromatographies here, like gas chromatography, and basically that is what is used mostly, of course, in pharmaceutical quality testing, also in research and development, is of course liquid chromatography. So some information about stationary phases, different ones, the chromatographic column, also how the apparatus is set up, what's the mobile phases, as I mentioned, how the apparatus is set up, how the instrument is set up, talking about gradient elution there as well. And then very, very important also, some general requirements that you see there mentioned as well about chromatographic columns, again, about system suitability, some basic aspects there. And what is very important is the adjustment of conditions, because it is very often the case that when you look on the method in the monograph and transfer it there, you are not able directly to fulfill the system suitability test there just by using the method as it is described. In the adjustment of conditions, it's very important to know that you are allowed in certain ranges to adapt the method, to adjust the method on the mobile phase a little bit, on the flow, for example. What else? pH of the mobile phase, for example. When you have a gradient running, you can also adapt a little bit and adjust the different phases there and percentages here. So that's very important to know as well. And still the method is considered validated there, and you don't need to revalidate that when you stay in that ranges. That is the real impact that this chapter on adjustment of condition, or subchapter in the chapter 6.1 is making. So that's a very important chapter, and that's why we use that one. And you see that is a chapter below 1000 and mentioned and referred to in the monograph, referenced in the monograph. Here you see an example for a chapter above 1000, giving some general information that is state of the art, what regulators are thinking, what very often the ICH is thinking. But very often also with our general chapters, we are preparing decisions that come later in the ICH level, for example. That was the case when USP back in the 1970s and the 1980s set up the validation chapters in USP 1225. That was used as the model then later for what is now ICH Q2, for example, the guideline there. The same happened about residue solvents, for example, which is now the Q3C guideline, and also for elemental impurities on heavy metals. There we had chapters already that were later then used very often in order to set up the Q3 guideline on the elemental impurities there as well. So that's also very often what is happening here. Also, of course, it works the other way around, but there is a fruitful discussion between the different organizations and institutions in order to bring pharmaceutical development further here. So that was about the documentary standards, reference standards development. I mentioned we do the collaborative testing. There's a work protocol that is designed by reference standard scientists there and approved by their peers and managers. In the document, in the work protocol, it's defined how many laboratories should participate, how many replicates they should do, if they should use just one analyst on different days, or several analysts on different days there, and other factors. And all that is put in the work protocol on a case-by-case basis. It always depending on how the reference standards under development is used there. The collaborators there thoroughly characterized each material using the work protocol and using then also compendial methods, but also non-compendial methods, especially for identity testing. They are using also additional non-compendial methods to get the identity right there. Collaborators may or may not perform the same test. There are some tests that are just done in one laboratory, our vapor absorption analysis, to see how a reference standard is reacting to different moisture environments, is basically done just in one laboratory, but the methods that are used by the 100% method to assign a value to a reference standard that is used quantitatively, this is done normally in all the collaborating laboratories here, in order to have a very robust result that can be used then by all the stakeholders then later. Each laboratory is generating their data, is reviewing their data before they give the results then to the reference standard scientists, and they are looking again, is there an agreement between the tests from one laboratory there, but then there's also, and much more important, is there agreement between the different collaborators, between the 3 to whatever laboratories are participating there, so that the values are not scattered too much, but that they are quite close together. And there are also specifications for that when we need additional testing in order to clarify issues here. I mentioned already that we use the mass balance approach to assign a value for quantitative standards. Mass balance means basically we have 100%, we are subtracting there the amount of organic impurities, the amount of residue solvents, the amount of inorganic impurities, the amount of water, and what is left over there, that is the value that gets assigned to the reference standard for the quantitative testing. The laboratory scientists, the reference standard scientists, they are also generating the label text. All the data from all the testing, from the testing protocols, is collated into what we are calling a reference standard candidate evaluation package. And here stops then the work of the staff of USP, and that package is given over to the Council of Experts that you can see here. Once again, we discussed it earlier before. Our volunteers, and they make, based on the data that they are getting from the package, they make the decisions to release that reference standard then for the use with the monographs, as we have discussed already before. And I mentioned already, setting up the reference standards for the monographs, setting up the monographs takes some time. And we are looking also and listened also to stakeholders that are saying we sometimes need quicker reactions by USP. So, and in order to work on that and react quicker to responses of the industry, we have, in addition of the reference standards that we have just discussed, we have also set up a new product line since 2 years. We have now this new product line that we are calling Pharmaceutical Analytical Impurities. And these provide, together with the reference standards, a comprehensive solution for research and analytical needs. So you don't need those pharmaceutical analytical impurities in the monograph for the official method, but with the help of these pharmaceutical analytical impurities, you can much quicker verify the Pharmacopeia method that you have transferred into your laboratory, and it helps with identifying impurity peaks there a lot, and it's just a very good addition to the reference standard portfolio there, while they are themselves not reference standards directly. So they are reference materials that you can use in your analytical work, but we are not calling them reference standards to separate them from the USP reference standards that are going through that very complex process I have described. So these pharmaceutical analytical impurities, they can be used for developing, validating, and transfer analytical methods that are maybe even not in the Pharmacopeia described there. They can also be used to verify pharmacopeia methods. For example, we have now let's not go through that. We can maybe discuss that in the question and answers if there is time, because I need to come to an end here. They can be used to identify and confirm peaks by comparing the retention times and the spectra there as they are present in the reference listed drug and as they are arising from test studies here. It's also possible to determine relative response factors with that to make quantification of impurities easier. And you can also work then with methods there and have specifications in order to release active pharmaceutical ingredients and testing therefore impurities that are not listed directly in the monograph. And with that, I stop here. I also stop my sharing and give the floor to Nikhil for the remaining of the presentation.
Thanks, Christian. Give me some time to switch over to my. Christian, can you confirm if you can see my slide 20?
Yes,
okay.
we can see.
Right. So Christian set a nice foundation in terms of what a pharmacopoeia is and how it can be implemented. The next 3 to 4 slides, I'm going to talk about how pharmacopoeias can actually play a role in design of experiments in early R&D and not just at the manufacturing stage. So just to refresh what Christian also mentioned, a drug product is basically a finished dosage, which is a combination of a drug substance. So in pharmaceutical terms, we call it an API, which is the active ingredient responsible for the drug activity, together with another ingredient. So in typical case, it's an excipient. Whereas a drug substance is an active ingredient that has an intended pharmacological activity in terms of what it's supposed to be having a mechanism of action and for what kind of treatment. The reason we mention drug product and drug substance in the pharma manufacturing is from a pharmacopoeial aspect. There are specific methods that one develops and refers to in terms of monograph or a journal chapter specific for a drug product or a specific for drug substance. And based on that, you also have reference standards that one uses depending on what has been mentioned in the monograph. So this is a typical workflow of an R&D. When it comes to the actual research, you have the R&D lab that focuses and takes through the journey of a molecule characterization, the molecule development from early discovery to LI/LO stage. In that time, a lot of assays, a lot of methods are developed, they are characterized, which are eventually transferred to QC, who will refer to either a known pharmacopoeia or a known standard, a documentary standard, or even refer to some form of other reference standard. So what is happening in the field now is the information that is being generated by QC and even at a later stage, that is being fed back into the early R&D stage. The reason being, in terms of manufacturing, where you have specific monographs, you have specific methods for identification of raw material to identify and quantify the quality of the raw material that you will use as your drug substance, add the excipients to make a drug product, and finally release it. There are pharmacopoeial standards and respective tests done for each stage. Now, where the R&D stage comes into play is all the information that is generated at this particular stage gets fed back into the R&D. That is where the importance of R&D utilizing a pharmacopoeial standard or even having an approach of pharmacopoeia helps later on in drug manufacturing. So the typical elements of the current pharmaceutical development is first step is to identify what is your QTTP, which is a quality target product profile. So this is basically a overall perspective and a picture of what you want to identify, what kind of a delivery system, what would be the route of administration. So you have to define that under your QTTP. Once you have identified and defined that, you then further elaborate what would be the critical quality attributes that will help you with the parameters of each of these QTTPs. So what is a CQA, critical quality attribute? It's a physical, chemical, and biological property or characteristic that has to be defined within a certain limit range in order to have confidence of the quality of that particular analyte that you will be analyzing. And this is not a one-off. This has to be monitored throughout. If you remember the previous slide, it has to be monitored right at the stage of R&D plus also at the lot release. So I want to show an example here. So this is a typical monoclonal antibody development CQA in a pharma company. You have all these quality attributes. So this is what you mean by the quality attributes in order to measure the size and charge heterogeneity. You have to analyze the molecular weight, species of a monoclonal antibody, the low molecular weight species of a monoclonal antibody, as well as the other variants that come from the charge variants. So these are not all, these are just an example. So what I want to emphasize using this example is you have to identify all these quality attributes that will obviously relate to what you have defined in your QTPPs. Once you have identified the quality attributes, then you go into a design space, more from a design of experiment perspective, where you show a relationship between the process input, which is the material, the actual analyte that you will put, and what kind of parameters that you actually have to analyze and use, which come from the quality attributes, and what are the relationship between all these process inputs and the critical quality attributes that you have identified. Once you have done that at the R&D stage, then you define and establish a control strategy. So what is a control strategy? An in-process control of your input materials, which is the drug substance, the active ingredient, together with the excipients and intermediates. So some other materials that you will add in order to run your assay or in order to manufacture your drug product. Together with other containers and some other forms of delivery system that you use in order to manufacture a drug product. And to give you another example and correlate to a quality attribute, what you see here is a chromatogram of a monoclonal antibody, which is a USP's monoclonal antibody as a standard. So when you will be designing a method, when you will be establishing a method, you will have to first run a certain standard. That standard has to perform in a certain manner. So you can see that when you will, in this case, the monoclonal antibody of USP, you see a high molecular weight dimer at a certain 14 minutes release time versus a low molecular weight species, as well as your main monomer. So once you have set a method that correlates to your quality attribute in order to measure a high molecular weight species, you can then run your sample. So a typical control or a standard that is run in routine assays. But the difference is why this is more relevant in pharmacopoeia is the method that is this reference standard is associated with. You can directly take that method and run it in your lab and verify it instead of developing that method all by yourself and then validating it. So that's where it takes you in terms of an R&D where you will be developing your own method. You will have to validate that. So the GMP regulations in terms of validating a method or even establishing a method is test methods to assess compliance of a pharmaceutical article, which has certain specifications that must meet the accuracy and reliability. So how accurate? is that method? Does it identify the analyte that you have targeted? And how reliable is the method? So again, the perspective is to develop and establish that method. Next comes the validation part. So any of the pharmacopeal method that you use, whether from a monograph or from a compendial chapter that Christian mentioned, less than 1,000, you can directly transfer that to your lab, use that particular method that is mentioned in the pharmacopeia, and you just have to verify it. Whereas when you will be developing your own method, you will have to validate it. So that's the difference and the value of having a method that comes from a pharmacopoeia where you don't have to validate, you just verify it. But when you don't have a pharmacopoeial method and you are establishing your own method, then you will have to validate it. So there are certain requirements in terms of how you validate a method. And these are the typical characteristics that you will have to look for and establish when you develop a method and you will validate that method. So how accurate is that method? Does it identify accurately the analyte that you're looking for? How precise it is? Does it pick up the analyte at certain concentrations? How specific it is in terms of having some other analytes. So for example, if you are identifying a monoclonal antibody, it will have other forms of dimers or even a low molecular weight species. So how specific is the method to identify that main peak of your monomer? Again, does that method that you have established, what are the limits of detection, as well as can it quantify low as well as a high quantitation limit? Then there are statistical tools that one has to use in order to establish that method. There are different models depending, again, what analyte you're analyzing, and then you use the right appropriate models in terms of statistically validating the method. Then comes the range, which will come from your detection limit. And then last one is how robust is your method. So there will be variables when you will transfer a method from, say, one lab to the other, or there will be variables coming from one hand to even another hand, another scientist using it. So does your method, is your method robust enough across these different variables? So these are the considerations that you will have to consider in terms of establishing and validating a certain method. Then lastly, all of this information that Christian and I have been talking, we have a very good plethora and catalog of courses that we offer. So they are all online courses. We can share a link after this presentation. So they are categorized in the drug products and APIs, biologics, food ingredients, as well as GMPs and excipients. So I would strongly encourage you to go and have a look at those courses. A lot of them are free of cost. So that will give you a much more embedded and in-hand and hands-on, kind of a training in those kind of courses. And that's it. So just to summarize quickly, I also want to leave some time for questions. The pharmacopoeia are focused on quality that effectively decide the safety and in the larger purpose of a public health. They are strongly embedded in the science, what Christian was explaining, through multiple labs and in collaboration with various labs, so not just in one lab. So that really gives you a collaborative and different platform-based results of the reference standards. The pharmacopoeias follow a transparent process so that it is independent, and anyone can come and verify whether it's done in the US or in any other lab. So that is the value of using a pharmacopoeia across the globe. And the standards that Christian was talking about, documentary standards as well as physical reference standards, they are established to support and optimize the method. So again, pharmacopoeias are responsible and they are more specific towards quality standards. So what kind of analytical development and analytical methods that you're establishing in your lab, those standards and those documentary standards can help you with those formations. So with that, thank you.